Hello, welcome back. So, in the last class, we have discussed about the protoplast fusion, about the fusogenic chemicals. In today's lecture, we are going to discuss about the somatic hybridization. So, first step, as I already told about the isolation, first we have to select the parent plant. So, in this case, this is the potato plant, that is the Solanum tuberosum, and the tomato plant, that is the Lyco persicum, that is the parent plants. Then you need to isolate the protoplast. Okay. After the isolation of protoplast, we need to fuse it by the help of the fusogenic chemicals. For example, the polyethylene glycol, the calcium, the sodium nitrate, right? We have already discussed about all these things. So then the protoplast fusion occur, and this will lead to the heterokaryon. It doesn't contain the identical nucleus. So homokaryon means which contains the identical nucleus, plating of the fused protoplast, then the growth of the hybrid colonies. Then this one is the selection of the hybrid. So we have discussed about first the isolation, then about the protoplast fusion, that is the second step. Then third step is the selection of the hybrid. Okay. So in the selection of hybrid, first when the hybrid forms, then the growth of hybrid colonies, then the selection of hybrid colonies, plating on the medium, then it will form a hybrid callus, and that callus we need to transfer to the nutrient medium to get the whole hybrid plant. In this case, the whole hybrid plant is tomato or the tobacco. This is the hybrid plant. So this is the schematic view of the somatic hybridization. So how will identify and select the somatic hybrid? So the identification is based upon the pigmentation, the cytoplasmic markers, that is the Fi tissue fits and the reds, that is then the presence of the chloroplast and the nucleus staining. And about the hybrid selection, after the identification to select the hybrid, that is by the genetic complementation, then next by the phytotoxins, next by the antibiotics, and next by the herbicides. So I'm just taking the example of antibiotics, how the antibiotic is helping in the selection of the hybrid. Okay, so let's take first one species, that is tobacco species, that is Petunia hybrida. Okay, we need to isolate the protoplast, right? And in this case, one more, that is Petunia parodi. Okay, same, the protoplast isolation. Then you need to fuse it. Then you need to culture with actinomycin D. That is the antibiotic. Okay. So in this is parent 1 and this is parent 2. So this parent will form small colonies, and after that there will be no further growth. So further growth ceases. Okay. And in this case also, in the parent 2 case, there will be no division and no growth. But when the fusion occur, and that fusion is cultured in the actinomycin D antibiotic, then it will produce large colony that will give rise to callus for the division. Then the last is somatic hybrid, that is desired one. Okay. So we can say that the parental species fail to produce the colonies and the division and the growth ceases. But when the hybridization takes place and the culture is with the actinomycin D, Then there will be the formation of the large colonies, and in the culture the callus will form, and that give rise to the somatic hybrid. So next comes is the type of hybrids, that is already I mentioned, that is first one is the symmetric hybrids, and one is asymmetric hybrid, right? So the symmetric hybrids is contain the somatic chromosome of both the parental species, and these are very significant as they show all the properties exhibited by the parents. Next is the asymmetric hybrid. These are those hybrids which preserve the genetic material of only one organism. So the chromosome contained of the other parent species is lost, just like the example of Oma. So next is about the advantages of the somatic hybridization. First, we have already discussed it helps in the production of hybrid, and that hybrid it could be the interspecific or the intergeneric. Okay, we have already discussed, and the intergeneric example is the pomado, which I already explained. Advantages of somatic hybridization. First is the production of hybrid. Second is the production of fertile diploids and polyploids from the sterile haploids. Okay. Then third is the we can transfer the genes, transfer of genes for selective purpose like the disease resistance or the stress resistance. Then the production of hybrids of nucleus and the nucleus and cytoplasm hybrids. Then here the molecular markers are also involved like the RFLP, restriction filament length polymorphism, then RAPD, then ISSR. So these are helpful in the detection of variation. And the last is the production of mitochondria and chloroplast hybrid, that is called the recombinant organelle genome, the mitochondria chloroplast fusion. So this is all about the somatic hybridization. I hope it makes sense to you. So if you like this video, hit the like button, share it with your friends, and do not forget to subscribe to this channel. Thank you.